So today we're doing a short review, which is a little odd for AP Bio, but we've squished an awful lot in a very fast period of time. I spent 3 screencastifys on functional groups and your macromolecules. And if you go back through those slowly, after each one I tried to review a little bit because I did go through those fairly fast. And then we spent 2 classes on organelles. One, a complete review of things that you should have learned in CP Bio, but you may not remember. And another class on that. One was focusing in on what the AP College Board wants us to pay specific attention to as far as an organelle. And the next class as a generalized review. Remembering that the AP Bio would like us to remember more, not just a brief description of what the organelle does, but also how it functions within the body. What is its role within the body? What happens if one of those organelles is damaged? So if I were you, I would want to know exactly what would be on the first test. And it's actually fairly straightforward because, again, we will spend time throughout the entire course on functional groups, the macromolecules, and the organelles. So this is just a baseline for us to establish when we're going forward. And I've told you the only functional group that I would require you to know on this test is the hydroxyl unit. So you would probably have to identify that or circle it or write that on the test. And then as far as the macromolecules, you should be able to list what the 4 are. And you should be able to give me the subgroup from that. For example, lipids, that there are fats, lipids, and the steroids. And then in carbohydrates, the simplest form is a monosaccharide. I would say that you probably should need to know that to form a disaccharide, we need to go through dehydration or removing of water. And to make a polysaccharide, we would keep doing the same thing, adding more monosaccharides to that and through a dehydration. For our proteins, remember that we have sort of a way that there are 8 different functions of our proteins, so they're very important as far as the any biochem course or anatomy course is considered. And you can figure out any way you need to remember all 8 of those. But for this course, for this first exam, you would be expected to be able to list what the 8 are, not their function, just be able to list them. And again, you might not want to use the shred CT. You might want to come up with something else for yourself. But remember that the S is for structural, the H is for hormonal, R is for the receptor, E is for enzymes, D is for defense, remember our antibodies, S is for storage, C is for contractile, either for as a muscle or as a contractile vacuole, and T for transport. And then the nucleic acids I spent probably less than a minute on. We have 2 that will become, that you would be expected to be able to identify in the course. And I would say that oftentimes when a student has been given, 100-odd days of college material and they are asked a very simple content question on the AP exam, oftentimes they get this wrong. When they're asked, can you identify a nucleic acid? I've seen so many times students in the past when I was a reader to leave a short answer blank because they forget it's as simple as DNA and RNA. And then we went over quite a few of the organelles, the ones that the AP College Board wants us to focus in on is what happens if there's an error in those. The endoplasmic reticulum, remembering that there is a smooth and a rough. And in that smooth, we had 3 big functions to that. It's where lipids are made. It is where calcium can be stored. And it is part for our, for breaking down or detoxifying. And then we had the rough ER, which is where the ribosomes are. And remember that the ribosome is the site of making proteins. And what is probably new to you is that endoplasmic reticulum is what they call, has a lumen, or it's like a tunnel. So things are manufactured and there'd be channels to allow those proteins to leave. And once those proteins are left, then they would have to go, for example, to the Golgi apparatus if they needed to have refolding or if they needed to have other molecules added to it. The lysosomes are very important in that they are responsible for, they have three large functions here. One is that they have digestive enzymes in them. And remember that if a person is missing genetically one of the enzymes, then they will have a genetic disorder. And I gave you the example of Tay-Sachs, where the enzyme is missing here. And remember, lysosomes have anywhere from 50 to 55. We know that there's at least 50 digestive enzymes, but it is believed that there are many more. So if you are missing one, you could have the disorder Tay-Sachs, for example, and in that case, missing the enzyme that would break down lipids in the brain, and the child, which appears perfectly normal up until the age of 6 months, the brain then starts to degrade. A second function of that, besides the enzymes, is that it acts as phagocytosis, or if there's material inside the cell that the cell needs to get rid of, and it's large, the lysosomes can actually break that down so that it's easier to get rid of in the cell. And its third function is for apoptosis or cell death. So if it gets the signal from either communication piece or another cell that there's something wrong and it needs to destroy itself, the lysosome can set that into action. The ribosomes, as we've talked about, are the site of where proteins are made. Remember that when the DNA is opened up and RNA comes in and reads the code, it only needs a specific portion of that code to make the amino acids to make proteins. But that code would go to the site of the ribosome. And that can happen either on the endoplasmic reticulum or the free-floating ribosomes within the cell. And what is unique about the ribosomes is everything that is living, every organism that is considered to be living, has ribosomes. So that comes into the evolutionary component of the course. So ribosomes are one of those organelles that you're going to be able to trace and say that has an evolutionary component because every organism has those. The Golgi apparatus we just talked about just a moment ago, or I talked about again, is that once that protein is made and it needs to be folded and maybe needs other functional groups added to it, that would happen in the Golgi apparatus. And remember that we have 3 types of folding. And you would be asked up here, you should be able to discuss the types of folding. And I'll go over that in just a moment. And then we have the mitochondria, and what we did not bring up during the lecture is that not only is the mitochondria the site of ATP production in the cell, or the energy for that cell, please don't call it the powerhouse of the cell. Remember, we're trying to get away from things like the brain of the cell or the powerhouse of the cell. AP College Board really doesn't like that. Say that it is where energy, ATP, is produced, or one of the places ATP is produced, because remember that ATP is also made in glycolysis outside the mitochondria. But another thing, another evolutionary component is that mitochondria only have the mother's DNA, and so it is the maternal DNA in mitochondria. So that is in both plants and animals. So again, that goes back to that evolutionary component that, for example, the ribosomes have. And I've mentioned these here. You would be expected to know at least 14 organelles in a combination of plant and animal. So remember that the plant has all of these organelles, but also has some unique in that it has the chloroplasts, that is a double-bound membrane where photosynthesis takes place. They also have the amylioplast that is for storage of starch. We have the desmonomatas that are for the cell junctioning, where the 2 plants are held together. It also have one large central vacuole for water storage. So we could also put vacuoles down here. We could put the nucleus down here, the nucleolus, etc. And let's go back to the types of folding with this protein, because that will also be expected on the test. So we have primary, secondary, and tertiary, which are your more common. We also have quaternary. And that primary is just a single strand of amino acids in one fold. In the secondary, we have 2 choices, alpha or beta. In the alpha, it is a helix held together by hydrogens, just like DNA is held that way. DNA is a protein that is held together by the helix hydrogen bonds. And then we have tertiary, which is, oh, excuse me, I did not go over our fan, our beta. Our beta also in our secondary would be folded like that fan, held together by hydrogen bonds. And then our tertiary would have layers and held together by our, or what makes it unique are the R groups or the side groups that are together. And again, if you have questions as to what is in the test, I would take a look at this. It's going to be very straightforward. Again, you would need to know only the hydroxyl unit for your functional group. You should be able to list lipid, carbohydrate, protein, nucleic acids, and what the subcategory of that is. Fat, lipid, steroid. You would not need to list any examples. You should be able to say monosaccharide, disaccharide, polysaccharide. And the way you go from a monosaccharide to a disaccharide is taking 2 monosaccharides, removing water through dehydration, and you would get a disaccharide. You should be able to list all of the 8 types of proteins: structural, hormonal, receptor, enzyme, for defense, storage, contractile, and transport. You should be able to discuss the 4 types of folding: primary, secondary, tertiary, quaternary, and the differences between them. This is one of the first ones that you're getting any other added information other than basic be able to list. So again, you'd be able to describe that in primary, it's a single fold. In secondary, you have either alpha or beta, alpha helix, held together by weak hydrogen bonds, or beta is pleated by weak hydrogen bonds. And then tertiary with your R side groups being unique to them. The 2 types of nucleic acids, and then be able to list and describe briefly organelles, whereas ordinarily I would expect probably what I'd say briefly is not one word, not to say ATP or a pathway or mixed protein. I would expect a little bit more of a sentence. For example, with endoplasmic reticulum, I would say that it has a smooth portion where lipids are made, where calcium is stored, and it is for detoxifying, where smooth is where ribosomes are made. Let's change this a little bit. Let's make this just 10. But having said the 10, you must include the ones, you must have the ones that the AP College Board say are the most important. And I would go back to lecture 2, it's actually the 4th lecture, but called lesson 2, to review specifically what those are. And if you have questions, I'm just an email away. Thank you.